Thank you very much. I want to thank the AACR and Dr. Szabo for inviting me to participate in this session. I have no disclosures. I derive my salary and my support from the NIH, the NCI, and the Fred Hutchinson Cancer Research Center. And the opinions I'm expressing are mine and not necessarily those of those organizations. Barrett's esophagus is a condition that we work with, and it's a condition of our era. In 1950, Norman Barrett described the columnar-lined esophagus, and he thought it was due to a congenitally short esophagus. In 1953, Allison and Johnstone saw more patients, concluded it was a complication of symptomatic gastroesophageal reflux disease. Barrett, to his eternal credit, 4 years later, reviewed all of the data and retracted his hypothesis and agreed with the new hypothesis. And in 1976, Nave completed the triad when he linked reflux disease, Barrett's esophagus, and esophageal adenocarcinoma in a large clinical series. And this paradigm has dominated clinical thought for more than 3 decades. It's led to endoscopic investigation of symptomatic reflux to find Barrett's esophagus, histology-guided endoscopic surveillance of Barrett's esophagus for early detection of cancer, and intervention typically for individuals with high-grade dysplasia or esophageal adenocarcinoma. And as a result of this, Barrett's esophagus is probably the best characterized example of the natural history of what we call a pre-malignant condition that we have. The difficulty is that we found that there were unexpected findings, and we have a paradox. If we try to base it on the paradigm where you take people with symptomatic reflux, find Barrett's, detect cancer early to try to lower the mortality. What have we found? Well, a large proportion of individuals with Barrett's esophagus are asymptomatic, and in a few minutes I'm going to try to convince you that that's due to their Barrett's esophagus. Nearly 50% of people who develop an esophageal adenocarcinoma do so without symptoms associated with reflux. So the patients don't know they're at risk. 95% of esophageal adenocarcinomas arise in individuals without a prior diagnosis of Barrett's esophagus, even though they almost certainly have Barrett's esophagus. Nearly 80% of esophageal adenocarcinomas arise without a prior diagnosis of reflux disease. So the providers don't know they're at risk either. Yes, we can screen people with reflux, but what we find is that there's no evidence that we can detect Barrett's esophagus prior to the onset of esophageal adenocarcinoma. And in these circumstances, we do not detect Barrett's esophagus, although it is almost certainly there. The patients present with a late-stage esophageal adenocarcinoma that has a very high mortality rate. Yes, we can screen and we can detect people with Barrett's esophagus, but we detect a benign, very indolent subset that almost never progress to cancer, and 95% die of causes unrelated to their Barrett's segment. So we can detect Barrett's, we can detect rare esophageal adenocarcinomas and do something about curing those, but we cannot do anything about the overall mortality in the population. And I bring this up because this problem of underdiagnosis of life-threatening early disease and overdiagnosis of early benign changes that will not affect the lifespan of an individual are rampant at every disease we talk about at this meeting. This is a problem that we must solve because we cannot cure cancers or prevent cancers that are never going to develop. Now, advances in the Genome Project have brought this, our study, which is now in its 27th year, to the point where we ultimately wanted it to be. We had the hypothesis that cancer is an evolutionary disease, and it occurs at the level of the organism, where the constitutive genome has been evolving over eons, and it's been studied by GWAS and will soon be studied by next-generation sequencing. And in this case, somatic cells curtail their function to benefit the individual, and they perform highly specialized functions to benefit the individual. The intestines absorb things. The heart pumps, for example. We're studying a different genome in clonal evolution. This is the Neoplastic genome. This is the genome that's being studied by the Cancer Genome Atlas. And this genome exists in space and time. The y-axis is a clonal expansion within the Barrett segment. The x-axis is that clonal expansion occurring over time. These cells enter selfish cell proliferation, where they lose the specialized functions. They're trying only to divide. They form a cancer, and that cancer is resistant to therapy. Well, this has been going on for a long time. These genes have been evolving for over 500 million years, the ones that we're finding in cancers. There are two strong peaks of these cancer-related domains. One was the origin of the first cells. These are what we call caretakers nowadays. They promote stability, and that was probably their role in the original single cells. But with the evolution of multicellular metazoan organisms, a second peak came into existence. This is the peak that controls and encourages cooperation among cells and prevent expansions of cheaters who want to undergo selfish cell proliferation, and we call these the gatekeepers. Gastric acid, we've been evolving with that for 350 to 400 million years. It's ubiquitous among all vertebrates, with the single exceptions of the monotremes in Australia. And it's a very dangerous experiment if you stop to think about it. Walking around with a bag of pH 2 hydrochloric acid inside your body, a lot of defenses have been evolved for that in the stomach and duodenum, which is exposed to the acid, and there's an incredibly rich network of things to try to keep the acid in the stomach and out of the esophagus. Yet, out here 400 million years later, in the last 40 years, we have an epidemic of esophageal adenocarcinoma, not previously reported prior to the 1950s. First noted in the United States, then in the United Kingdom, then in Western Europe, Australia. We're now seeing troubling signs from Asia, where it was previously unheard of. So it's becoming a pandemic in 40 years. And that means something has changed in the environment. And these are what we believe have changed: obesity, tobacco use, reflux disease, and a diet high in fat and low in fruits and vegetables are believed collectively to account for about 79% of these cases. So overdiagnosis, what is it? Why do we always get fooled? What's going on down there? Well, Barrett's, we're starting to learn what that is. Barrett's is well-differentiated epithelium in the distal esophagus, shown on the left, is being bombarded with acid, so it secretes bicarbonate. And that's one of the reasons it doesn't have symptoms. It's being bombarded with acid, bile, and tobacco products, so it secretes this thick, adherent mucus that covers the cells and provides a protective layer. The pain comes when the cells are damaged and the acid goes out into the paracellular spaces. Barrett's has special tight junctions made of claudin 18 and other claudins that are resistant to that acid degradation of the epithelium. It overexpresses genes involved in defense and repair. It maintains intracellular pH in the face of prolonged, repeated reflux exposure. And most importantly, it has a well-differentiated crypt epithelium where the stem cells are buried at the base of the crypt under all of these protective layers, and if they get mutated coming up, that mutation gets sloughed off into the lumen. And all of these are adaptations that the Barrett's has picked up from other cells in the GI tract that are being exposed to acid and bile. And so the real question is Barrett's esophagus the fire truck at the fire? And we're misinterpreting it when we call it a pre-malignant condition, and that's the reason that it exists for the lifetime of most of us without ever bothering most of us. Well, why do some people go on to get cancer in Barrett's esophagus. Well, the clue for this was discovered nearly 20 years ago by a man named Doug Levine. We were working together. Both of us were young assistant professors at that time. I was studying the genetics of Barrett's esophagus, and Doug was studying transmission electron microscopy. And he discovered this mucus secretion, and he correctly noted that it was coming from cells that were very similar to cells in the stomach and the duodenum. And we put our work together, and what we found was that in the patients who had entered a chromosome instability pathway, which we measured in those days by DNA content flow cytometry, aneuploidy and tetraploidy, that pathway was destroyed. We typically went through the Golgi and the rough ER, and to mucus granules that got secreted, and the cytoplasm was simplified, the Golgi was extremely abnormal, and they were not secreting mucus anymore. And so Barrett's esophagus is an adaptation to its environment. And it arises in an environment of chronic reflux, local and systemic damage from acid, bile, and tobacco products, and inflammatory responses to all of these injuries. And all of these exposures are mutagenic and known to cause chromosome damage. What we're doing now is a study in Barrett's esophagus where we measure all of the exposures, including a GWAS in the oval on the left-hand side of the screen. We're measuring the systemic and local toxicities with metabolic syndrome, local inflammatory reactions in the blood. And then in the yellow, we're doing a case cohort study of 250 patients, 80 of whom have progressed to cancer, and the rest have not progressed to cancer. We're taking one biopsy every 2 centimeters in the Barrett segment, and we're using the Illumina OmniQuad array, 1 million SNPs, to measure at the first endoscopy copy gain, copy loss, LOH, and breakpoints, which is the baseline mutational load that these patients have in their esophagus. And then at the penultimate endoscopy, either before the cancer or the next to the last endoscopy, we repeat those measurements, divide by time, and get a rate, which we call somatic genomic instability. With the advances in the Genome Project, it's now possible to study this evolutionary process in space and time. These are studies we did in the 90s. The only way we could do them was the Genome Project was uncovering microsatellite polymorphisms so we could do LOH. What you have here is an esophagectomy specimen. The top is squamous mucosa, the bottom is gastric mucosa. The dark dots indicate a cancer arising in the precursor lesion. The light dots, high-grade dysplasia. The high-grade dysplasia had inactivation of P53 by LOH and mutation, and 3 different aneuploid populations. When the cancer evolved, it had 2 more aneuploid populations and a 5q LOH. The Hopkins group in colon cancer and pancreatic cancer has been publishing what they call comparative lesion sequencing, which is a spatial way to get at things, including estimates of mutation rate. So when we completed our spatial scale experiments, we had done a genome-wide scan for LOH, which was state-of-the-art at the time, and we had 5 regions that were selected above background, and the rest we thought were neutral regions. We had done analyses, like I just showed you, to find out what abnormalities occurred before the cancer, because our priority was early detection and prevention. And so we wanted only those things that occurred before cancer. And that boiled down at that time to 4 things. 9-PLOH, which we knew at that time included the P16 gene, 17-PLOH, the same for P53, and the DNA content flow cytometric abnormalities. And based on the spatial scale experiments, we inferred an order in which these occurred, and that was subsequently validated in prospective studies. And then we put them all together in a biomarker panel in a 10-year prospective cohort study. These were 243 patients with Barrett's esophagus with intestinal metaplasia, followed for a median of about 6 years at the time we broke the study open. The biomarkers were the ones we had identified from the spatial scale experiments. The outcome was esophageal adenocarcinoma. And when we looked at the data, the gene-specific events didn't matter. They didn't provide independent risk prediction. What mattered was the chromosome breaks, the LOHs, the tetraploidy, and the aneuploidy. And these were the things that were conferring the risk on the progression to esophageal adenocarcinoma. When you looked at the results, we characterized the patients at baseline and then followed them, some for up to 10 years. If they had all three of the abnormalities shown in the title, they had a 79% 5-year cumulative incidence of cancer. If they had none of the abnormalities, they had a 0% cumulative incidence of cancer out to nearly 8 years, and there was a sort of dose-dependent response with the acquisition of more abnormalities. Tom Vaughn, we've collaborated with for years, is interested in NSAID use. The NSAIDs being measured here are aspirin, largely 81 milligrams, and Advil for sports medicine injuries. And what he found is that people who are current users of NSAIDs have a lower rate of progression to cancer, but they also have a lower rate of progression to develop aneuploidy and tetraploidy in follow-up. So somehow it's influencing the incidence of those events. Now, one thing I want to take away from here is that clonal evolution is a dynamic stochastic process. And what that means is we're never going to get perfect biomarkers. You can never get that ROC curve all the way up to the left because the evolution is dominated by outliers with increasing fitness that are selected and then undergo clonal expansions. The biomarker is based on the presence of an abnormality in a population of cells are inherently unstable. They may not yet have evolved, or they may be only a minority population that's waiting for selective pressures to change. Based on Tom's data, we sort of were interested, well, what happened to these high-risk patients if they took NSAIDs, current use of NSAIDs? And so we said at the time we diagnosed them, we looked, and people who were the highest risk, those with 2 and 3 biomarker abnormalities, we divided them into those who were using NSAIDs. And those who were not using NSAIDs had an 80% 10-year cumulative incidence of cancer. Those who were current users of NSAIDs dropped to 30% or less. There was a trend in the lower risk patients, but it was not statistically significant in a study of this size, although it might be in another study. Now, we usually present this as a success, but the people there under the black arrow, it wasn't a success for. They got cancer anyway. But we can learn from that if we're taking biopsies in our cohort studies, because if there's a mechanism for resistance to aspirin or other NSAIDs that allows them to evolve to cancer, we can learn from that and put it into our prospective studies and our randomized trials for monitoring them. So again, the implications for cancer prevention, neutral mutations matter. When we did our original studies, we only studied selected mutations, but interventions like we try to use for cancer prevention can change the selective pressures in a neoplasm. And by identifying these changes before we get into a randomized trial, we can identify them early and identify the patients who are at risk for progression, even if they have not yet progressed. And so one way to go forward is to look at the genome rather than genes. And that's what we're doing in our study. And one of the most fundamental evolutionary properties is the mutation rate. And that's what's being worked on in the comparative lesion sequencing in the Hopkins group. They're working on the point mutation rate. We're working on the chromosome mutation rate. And if you can modify that mutation rate, you can probably modify the progression to cancer. There are other things that are fundamental to evolution: the effective population size, which is the stem cell population, natural selection, which you can monitor by clonal expansions, as shown on the slide, and the generation time. Carlo Mele has been a pioneer in this work, and this is some of his work with us. And what he's looking at is the size of a clone and progression, and it matters what the clone is. If you have a genetically unstable clone, one that's lost p53, or an aneuploid or a tetraploid population, then the risk increases exponentially with the size of the clone, and you can see that in the Kaplan-Meier curves. But not all clones predict progression. The size of a CDKN2A clone doesn't predict progression unless once you control for p53. So just being a big clone is not intrinsically bad. Now, evolution operates on variants that it can select from, and genetic diversity is the fuel that is driving evolution, and it's driving the progression to cancer in the patients that we're monitoring. And this is a paper from Carlo. This is his first paper on the subject, and in this paper he measured 3 diversity measures, the number of clones, the Shannon index, which is basically the entropy equation, and genetic divergence, which is illustrated in this slide. He's recently, with Lauren Merlo, published a comprehensive list of diversity measures, which those of you who checked your email today got a copy of in the current issue of Cancer Prevention Research with a very nice editorial accompanying it. The good news for us is we don't have to worry too much because they're all significant predictors of progression. As shown here, just counting the number of clones gives you a marked increase in the top quartile of progression relative to the lower ones. And all the diversity measures are statistically significant, and they're all about the same, so we can use whichever one turns out to be the easiest for us to use. We're not going to be forced to choose between measures. Now, I realize that this slide isn't from the Cancer Genome Atlas, but the point I want to make is that cancer is more complicated genetically than any one of us imagined 10 years ago. And that's showing up in the Cancer Genome Atlas. It's showing up in this slide of a lung cancer, which has the little circle of dots surrounding this are 50,000 high confidence single nucleotide variants, point mutations in this cancer. And not to mention the major chromosome damage that's indicated with all of the various colors in this cancer. And so you can look at genes in this cancer, and a heck of a lot of them are going to be abnormal. And the genome is really, really abnormal. And the implications for this are several. One, when we did our study, we'd used flow cytometry and STRs. Now we're facing total genomic sequencing. A rapidly advancing technology overwhelms biomarker validation, and you have to get in front of that. You need to anticipate what's there and what you can get into the clinic really fast because the new technology will sweep you away otherwise. This requires that the cancer prevention field have a cancer precursor biobank that you can go to rapidly and validate. Just the same as they've been creating a human cancer biobank, we need a cancer precursor biobank. Now, the thing that's going to help us is that the Cancer Genome Project is going to, the Atlas is going to identify regular changes, and people are going to try to develop treatments for those. And then we can find out and incorporate those treatments, if they are low toxicity, into prevention strategies if we know what mutations are there in the earlier stages. And so we will get help from that project. The final thing I'd like to leave with you is that neoplastic evolution leads to progressive, irreversible loss of genetic information. And we don't know how to handle that very well. And this are 4 stages of Barrett's esophagus. Early Barrett's esophagus are patients who did not progress to develop aneuploid, tetraploid, or cancer over a 10-year period. These are the ones that you want to just tell them to go enjoy their lives. The second stage are patients who progressed to chromosome instability over the same period of time but did not progress to cancer. These are the patients you want to intervene on to prevent the development of cancer. The third are patients who developed a small, early curable cancer, and this is the Barrett segment that the cancer arose in. So this is early detection. The final is an advanced esophageal cancer, and what you see is a statistically increasing incidence of irreversible loss of genetic information as you go through this process. And the implications for this are we should try to prevent instability, and if we can't, we should take it out. So I didn't do all this work myself. I actually did very little of the work, and I'm always the one up here presenting. So I just want to note that this is a team effort, and there are too many people for me to read them off, but I'm extremely proud of all of them, and I want to thank you for listening. Thank you very much.
Thank you, Brian. The floor is open for questions. So, Brian, maybe I'll start out by asking you whether the genomic alterations that you describe within Barrett's esophagus extend to the histologically normal cells around it. That is there a field of injury, as we've described, as was described by Hemont?
Yeah, some of these will expand throughout the entire Barrett segment. Some of them are quite localized, but we reproducibly pick them up with one biopsy every 2 centimeters in the Barrett segment. I think the thing that's where the jury's out on is how fast they go to cancer, because that may be where we're getting the underdiagnosis. They may go to cancer fast relative to the time we look down with an endoscope. And that's what our study is trying to find out.